Okay, time is ready. And we are going to wait another 5 minutes just while chatting the wishes and hopes and also the progresses. Okay, so still I have checked the sites of participation and also the numbers of the participants, and more than the much happier and than expected. Then everyone could stay, even though we are still under a kind of strict, strenuous, and severe, and still the scanty of hopes for conquering the current situation. So here you are seeing my advertisement. Me and Professor Saul, and in long story short, and we can continue the correlative imaging, and also the next semester might be the proper time for brain. So to guarantee to just to keep the diversity in hand, and I wish everyone would recommend, advise us what might be the appropriate one. So not the next semester, but the next to the next semester, if anyone or everyone or many of you wish the topics of the thyroid cancer and treatment, though I'm not an expert in that field, I do not see the people with thyroid diseases. Last time they just took care of the thyroid patients were almost 25 years ago. So, however, I can just I and Professor Saul can they coordinate the lecturers who would be so far very much willing to give a lecture in English, and almost half of them are also agreeing with us that those lectures are going to be recorded and to be given for all the participants. Okay. All right. So today I'm going to complete this semester's lecture series, and then two of the participants who are also participating, they attending this lecture series as a part of their obligation to take the graded course seminar, and those two. Tomorrow I'm going to give the questions and send those questions to both of you, the regular attendee, and then as is the precedent, you are going to send me back the answers to those questions until the Tuesday next week, and then I'm going to just score them, and then you're going to receive the grades, and you're going to complete the requirement of the graded course. For all the other participants, they just also like the previous precedent, we are going to upload those questions mostly 1 or 2 from each lecture are going to be uploaded in our nmportal.org with the answers. So so you can just kind of take a glance at, and freely, it's kind of a Creative Commons. Those questions and answers were developed by each lecturer, and they also gave us the consent to for any of you to use them for later the study or later even further use if it is not for commercial. Okay. So I think everyone would have been still only less than 10% of the Korean people have been vaccinated. And in a hurry, our president had been in the States to beg for them to share the Pfizer or Moderna vaccines. But sadly, unfortunately, it was unsuccessful. It's because USAID is going to handle, and well, actually the US president and then the US government is handling the surplus of vaccines to be given to the low and middle income countries. And I know the situation of low and middle income countries are desperate. So I agree with the decision by the US president and also the government. And South Korea is one of the recently advanced, recently developed countries, and we needed to take care of our own. And anyway, it's less than 10%, and that's the reason why we could not start another KOICA project. But however, 10 project is online, so 10 project is going to be submitted again in June as a concept, and also in September, the next phase. Next phase is going to broaden the number of the countries like Nepal and Mongolia. Less than 10 experts are working, next generation leaders are working in both countries, and we are more than happy to share our lectures. And also the fellow of Asian Nuclear Medicine Board, FANMB, those people are going to be contacted by Professor Soo in an appropriate manner. I just discussed this issue with Professor Bom, who is the chairman of the ARCCNM recently on the phone, and we agreed and we are going to expand the audience. And so as you are the early birds to have been in this lecture series, and last year you have enjoyed the series of lecture by Professor Edmund Kim. And Professor Edmund Kim gave me a feedback that he felt like he came to be young again. Why? Because all those participants from every Asian country, you look so young and so eager to learn from him. And then you helped us to choose the appropriate lecture titles. And as I remember, that 30 years ago, the situation of my country, mine and my country, we could not keep up with the progress of the level of the progress and level and quality of nuclear medicine practice, and also the related sciences of the advanced countries like Germany, United States, and others easily. They just try to help us eagerly However, they have had their own business and their own things to do for the progress of the nuclear medicine. So after 30 years, we found that there has been a great success in the hybrid imaging like PET-CT and PET-MRI, especially for cancer. However, there has been a lag in brain. And interestingly, due to the unique situation of United States and in the States, 20 million, among 20 million cases of nuclear medicine procedures, which have been and which are being done annually, the 10 million, half of them are nuclear cardiology procedures, which is beyond the touch of the nuclear medicine physicians. And the American, meaning the North American, meaning the physicians of nuclear medicine physicians of United States, they do not know, they don't care of the nuclear cardiology. But for us, and for almost every one of you, in addition to thyroid cancer and bone scan, and the microspect and the heart cases are real pragmatic and practical issues of everyday routine. Then what about brain? Brain is an interesting field. In 1984, the first science science article was published by the late Professor Henry Wagner. Henry Wagner had been at the time the Johns Hopkins University, and he and his colleagues for the first time from the nuclear medicine field and published a science paper regarding the dopamine, the receptor imaging of the human, and also, as I remember, it was C11 methylspiperon, which is directed to the D1 receptor, but my memory is waning. If you are interested in the history of that, and you should be, you might be checking yourself. And during that time, during huge success of the hybrid imaging, there had been another progress of a small breakthrough from Germany. Interestingly, it is from Germany. You know, the modern nuclear medicine has started from Hungary. It was the George de Hevesy of the Hungary, Budapest, and then he was the one, and then his discipline had been transferred to the University of Freiburg, it's a southern German university, and then they propagated throughout the European continent and even to the States. So, you know, interestingly, Hungarian society, their name is George de Hevesy Society of Hungary. George de Hevesy Society of Nuclear Medicine of Hungary. That kind of issues. And also, we know that there is another big guy from the big shot from the Poland, and her name is Maria Sklodowska. So that's the reason why she later came to be the wife of Pierre Curie in France. So we know that the Madame Curie, but that Madame Curie's the maiden name is Maria Sklodowska. So the current president of the European Association is from Poland. And then previously it was Marie Curie lecture, but I'm not quite sure the lecture name has been changed or not. But fellowship of the European Association is Maria Sklodowska Fellowship. Okay, so that's the brain issue. So brain was the one which has promoted nuclear medicine physician to become the author of the eminent the science paper. But that Professor Henry Wagner blamed all of his descendants, meaning his offsprings or his students, ex-students, the 2011 in Japan. And in a seminar place, he expressed this disappointment and frustration because of the slow progress of the clinical application of the brain nuclear medicine. And during that time, the theranostics were in a rush pioneered by the Professor Richard Baum. And Richard Baum had made a small breakthrough, and which is now the mainstream of the Nuclear Medicine Society, which is the correct direction. So that's theranostics. But everyone thinks that the theranostics is just kind of one of the small discipline of therapy or oncology. Nuclear oncology has expanded to theranostics. Who can let me just handle the echoing issue permanently. I'm going to just make okay. While just consuming several minutes, actually 10 or more minutes, while just roaming around our history and then current status, and now we arrived the issue of the brain theranostics, which subject I'm sticking to, especially for during the rest of my life. And then today's lecture title will be update of the brain theranostics. And actually, this is an introduction of next semester. Next semester lecture series is going to contain regardless of your the advice or recommendation, and next semester's topics would include Alzheimer's disease, amyloid and tau imaging, by Professor Yu-Kyung Kim, who had been Johns Hopkins for 2 years, 20 years ago. Now she's a chairman of the Metropolitan Hospital, which is affiliated with Seoul National University too. And also I have invited, and he willingly, happily accepted my invitation, Professor Jason Kim. Jason Kim is the renowned expert in Parkinson's disease. Then he's from the Asan Medical Center, and he's going to talk about the Parkinson's disease. And also, if you allow me, and I'm going to invite several PhD scientists from my colleagues or from all of the country of South Korea. And if you wish, then we can just teach you, or we can let you know briefly and let you browse the progress of the current use of, for example, the statistical parametric mapping, SPM. I don't recommend the use of the program of Professor Yeah, so I forgot the name of the program, but you know the amyloid imaging and tau imaging, those images are on the on his program, but this is unique to the Nuclear Medicine Society. MRI and fMRI and other fields are using SPM, which was mainly developed by the University College of London and Professor Carl Freestone 26 years ago. And I was one of the crew, one of the participants to use that. And it is trivial now. And we are moving on to the terms and connectivity studies, which is still beyond the use of and use of the clinical purpose, use for clinical purposes. So today I'm going to talk about the interesting and unique title of the brain theranostics and update. And this is the continuation of my previous lecture, my previous presentation in September last year. And at that time, I just put the title Flow Physiology, continuing the myocardial the SPECT story, Flow Physiology and acetazolamide brain SPECT. And at that time, then I just emphasized that our things are based upon the physiology of blood flow, myocardium, but we need to move on to the brain. And radiopharmaceuticals and physics issues and acetazolamide stuffs and difference between heart and brain in vascular reserve and when to use and AI supported reading. And I don't think I covered all of these items. I just tried to introduce to you the main the issues of these subjects. And today I'm going to move on a little bit. Today's topics and issues consist of the brain theranostics. I just used almost 20 minutes to explain the brain theranostics and then current and promises. And brain theranostic had been there. I just renamed them, and I'm going to edit the current and then recent history of use of the brain, nuclear medicine. And then there had been a change of understanding brain, which is remarkable. I'm not talking about the cognitive neuroscience. I'm talking about the molecular biology of brain and recent trend of the transcriptomics. And I'm going to explain to you PET and their promises. Let me remind you that especially Indonesian colleagues, when I had been in Bandung, the 2018, after a brief introduction, in a kind of group, and Professor Mashur had been there, and I just introduced my plan of the issue of the Trans-Eurasian Information Network, this project, and TAIN, and it was short, and it was in the heavy rain. So I remember that I needed to almost shout. So within a small room, if there were not the squall in the tropical the weather, I could just kind of gently speak, but I needed to shout. And all of a sudden, after the presentation and meeting, and rain stopped, and we gathered together. I remember those people and the young faculty from Hassan Sadikin, and one of you asked me as I just manifested myself as a brain, actually a brain specialist. So one of you asked me what might be the near future of the nuclear brain or CNS nuclear medicine or nuclear neurology or and psychiatry. So I immediately answered, it needs time to explain that, maybe 1 or 2 hours. So after that, I studied almost 2 years. I found that the 2 hours were not enough. And I was really behind to understand the recent progress of the brain. And especially after the COVID-19 crisis, I'm stuck in my country, in my office and home. So I studied almost a full time without being distracted and diverted to any other agenda or the business. So I'm going to let you know what I found. And that question was the initial point. Actually, it was a kind of a stimulus for me to explain. I had, 2 years ago, I had a vague concept or vague recognition of the current progress and the future. But for you, the next generation leaders from Asian countries, it would have been serious. And I have been disappointed by the failure of my colleagues in neuroscience. What I'm saying is just a global neuroscience, not to generate or produce any remedy, no therapy, and especially for neurodegenerative diseases so far. And then the critically ill brain patients are dying. And unlike oncology, we don't save the brain patients. We just kind of describe. I feel like the brain and especially the psychiatry or for serious devastating brain disease such as the subarachnoid hemorrhage or intracranial hemorrhage or brain trauma, for those patients, our neurologist and psychiatry colleagues they seemed to be Hippocrates, not a modern physician and surgeon. So the 2 or 3 years of honest study improved me, and let me just share my understanding, pattern their promises. And classic topography and connectomics are not going to be handled today. If requested, if asked, I'm happy to just to share my expertise. I'm one of the experts in this field globally. And abnormal protein imaging, and mostly tau and the amyloid. Those issues are going to be critically dissected, and we need to just find the bright side and as well as the dark side. Every lecture, each of them, used to emphasize the bright side. So there was the theme of the question from the Indonesian colleagues. Do we need to catch up with the progress of Australian and also Australian meaning the Christopher Roy and other the German and others the pioneering achievement in terms of the amyloid and tau imaging. That was the core question, which I could not immediately answer. Today, I'm going to answer to that question. And unexpected meaning, those abnormal protein imaging has, in my opinion, flaws, critical flaws. And to understand that those flaws of the abnormal protein imaging using PET needs the kind of a little bit of in-depth study of the neurodegeneration and new targets based on spatial transcriptomics. You are going to see the hope for the future. And because those the spatial transcriptomics is based upon the cutting edge technology and budgetary support. But interestingly, recently, a public database is available. And all those studies, all those the analysis, just like a topographic, the SPM or the connectomics studies, those spatial transcriptomics are being done on the computer. And you don't need to build a wet lab. You don't need to invent wheel again. So everything is there. So what we need is just to kind of understand the progress of the current one. The meaning, the current concept and the possibility to understand the brain diseases. Okay. I'm going to use the previous one again. Brain theranostics current. In case of the existing therapy, nuclear imaging guides therapy. One field is epilepsy. Epilepsy cure by corticectomy for neocortical epilepsy or hippocampal amygdalectomy for mesial temporal lobe epilepsy. There are two types of epilepsy. And by FDG PET and ICTAR SPECT. 25 years ago, we started a epilepsy board meeting. And after 10 years, actually 7 years, 1,000 cases were operated because of the skills and expertise of my colleague neurosurgeons in this hospital, this one institution. They operated 1,000 patients, and among them, 60 to 70% of the cases were mesial temporal lobe epilepsy, and among them, after operation, there are grade Engel class 1, 2, 3, 4, and you know the Engel is a neurosurgeon, and Professor Engel's classification. So 1 to 2A is improved or cured. And my neurosurgeon colleagues, they could achieve the 87%, and that's almost a world record. 87% of the patients improved. And once improved, then that's the absolute, the gold standard to indicate epileptogenic zone, like here. Left frontal lobe was an epileptogenic zone. Why? Because after successful operation, after 1 or 2 years of follow-up, that patient had been found to be cured. So 87%. And after 10 years and 10 or 15 years, we could just even the successful use of ICTAR SPECT and FDG PET to localize epileptogenic zone. And we now know that the long story short, when there is a hippocampal sclerosis or atrophy on MRI, there is no need to do ictal SPECT or FDG PET. You just do the operation and patients are going to be improved. However, we have seen bilateral cases. We have seen interesting normal hippocampal structure or amygdala, and patients are showing the typical mesial temporal lobe epilepsy type of EEG. Then those cases or bilateral cases or neocortical ones, mostly neocortical ones, we helped them to lateralize, to localize, and or guide their location of the grid and grid insertion to do the intracranial EEG to find out the location of the epilepsy, the seizure focus. Briefly, they said seizure focus. So during the third decade after the introduction of globally ictal SPECT and FDG PET, interestingly, in 90s and just after the millennium, we used popularly the FDG PET and ictal SPECT, and we are not using them very frequently. Why? Because all those easy cases are operated just after the MRI. So we helped to establish that kind of a scheme. And basal and acetazolamide SPECT for cerebrovascular diseases to find out the patients of the patients who are indicated for stenting or endarterectomy or surgery, bypass surgery. There are 4 types of operation for carotid artery stenosis, and there are differences between young and elderly. So getting rid of those kind of details, if I say in conclusion, it's just there are 2 extreme types. One is very young people and 3-year-old younger, 3-year-old or younger. Those younger children, the operation risk is high, but the letting them not operated with observation, the risk is higher. And they do need to receive operation. That's indirected bypass surgery. We call it EDAS, EDAS. The last word S is synangiosis. And that's one extreme case. Another extreme case is carotid arteries, the elderly. In elderly case, unlike the young children, carotid artery stenosis is caused by the atherosclerosis. Since 90s, which were published serially in New England Journal, and the most recent one in JAMA, Journal of American Medical Association, 3 times of the randomized cohort trial did not prove the endarterectomy or bypass surgery. In this case, a direct bypass surgery, which is direct bypass surgery from superficial temporal artery, which is a branch of the external carotid artery, to the middle cerebral artery, MCA, which is the branch of the internal carotid artery. That kind of bypass surgery or endarterectomy. Endarterectomy is endartery of the meaning the endothelium, endarterium. So endarterium of carotid artery of interest showing stenosis, and due to the atherosclerosis is removed after opening mostly the neck. And those invasive procedures did not prove superior to other methods. And still we are losing dozens to several dozens of patients because of the stroke. And after lucky, the recovery, they are experiencing neurologic deficit. So still, in those cases, we need to just to apply the Diamox-Spect, the brand name, commercial name of the vial containing acetazolamide is Diamox. Acetazolamide is too long, so we call it Diamox-Spect. And here is the triage point to epilepsy operation. Hippocampal sclerosis and atrophy, and mesial, they call it mesial instead of medial. Mesial temporal lobe epilepsy and HNA-ectomy. Cryptogenic mesial temporal lobe epilepsy, we do still ictal SPECT and FDG PET, and then HAN-ectomy. And also neocortical epilepsy, we do ictal HMPO SPECT and FDG PET, and subdural strip and grid, and ictal EEG, and then resection. From my institution, 2017, this non-lesional neocortical epilepsy. There are easy, the lesional neocortical epilepsy caused by what? Caused by the tumors or AV malformation or cortical dysplasia, which is explicit on the MRI. Then we need to do just kind of the operation. But in case of the non-lesional, it includes non-lesional. It includes the indolent, no. Inoperant, no. Latent, no, and whatever. So MRI normal. The incipient, no. So anyway, so they used the term finally non-lesional. There is no lesion on MRI. However, it might be shown here, PET. Then after subdural ictal EEG, we do the surgery. And surgical outcome after 10 years. Engel class 1, completely cured, is 60%. 2, and then still, so there was a marvelous the achievement. We helped them to achieve that goal. So JAMA Neurology, several years ago. So here, PET had helped. I'm so proud. So you need to find this kind of issue in your institution. And that's what I have summarized previously. In detail, extracranial atherosclerotic, extracranial meaning the neck vessel, atherosclerotic internal carotid artery. They call it steno-occlusive disease in the elderly. COS trial, they found no effect of EC-IC bypass, external carotid and internal carotid bypass surgery to prevent stroke. And CREST trial is no effect of carotid endarterectomy, endarterium, endarterectomy over medical treatment. ACT trial, stenting equivalent to endarterectomy. So maybe the neurosurgeons will be fired if they don't have any the item to help their patients. And interestingly, Far Eastern countries like Japan, Korea, China, Taiwan, and of course Hong Kong, and also Macau, and also Vietnam. There had been a report that moya disease in the young children and adult, very young, as I told, risky, but indirect bypass surgery is a textbook indication. And children definitely benefit with indirect bypass surgery. Yes. And symptomatic in the adult, of course, we need to just to do the direct ECIC bypass surgery. Why? In the grown-ups, the temporal external carotid artery branch, superficial temporal artery has a sufficient thickness, sufficient size, so which can be used for direct bypass surgery. And recently, people are trying to figure out why some of the asymptomatic patients are going to be symptomatic sometime. So they do use Diamox-PECT to find out who to wait for the right time, and who to operate immediately. Medical waiting for STA MCA bypass surgery. That's one slide summary. However, that Diamox-PECT study is not safe. During the last 10 years, Japanese colleagues, they experienced, in some cases, anaphylaxis like COVID-19 vaccine and acute pulmonary edema, probably due to, they concluded, metabolic acidosis or the cardiogenic. So metabolic acidosis was supposed to be derived from the inhibition of the carbonyl anhydrase. Metabolic acidosis meaning the blood become acidic due to the pharmacologic action of Diamox. So they have set up the guideline. The guideline is saying that in the elderly male smoking patients with a limited tidal volume of forced vital capacity, in those cases, or FEV1, forced expiratory capacity at 1 minute. 1 minute, or I forgot that. Anyway, FEV1 or vital capacity or tidal volume. In those cases, they recommended the monitoring the respiration and also the oxygen saturation and those issues. We have not experienced that. However, unfortunately, we have experienced a case 5 or 6 years ago, acute brain infarction in posterior cerebral artery territory, 1 month after surgery on Diamox-Chalin. During the acquisition, why? Because Diamox is given on the table within the SPECT scanner. So this girl had experienced this kind of acute edema and transtentorial herniation and brain death. And this girl was saved after the strenuous CPR in my department. However, after sending that patient to the ICU intensive care unit. They could not save her brain. And then after several months, she, according to the parents' decision, she kindly donated many organs to the recipients, and then passed away. So after that, they have reported it in the child's nervous system, and two independent group, a panel, had reviewed this case. We could not find any reason. It seemed not to be the drug effect, meaning idiosyncrasy, anaphylaxis, or anaphylactoid. And also, it did not seem to be the problem of any the artery. Why the details are here. So finally, both of the group of professors and the investigators, including me, we could not understand why. Recently, I found this kind of data here. In 2017 and 2019, and by the Swedish group and also the American group, JCBFM, and also Science Advances, in those prestigious journals, they say that the acetazolamide is just like a cisterna magna puncture type of, and there has been a kind of similarity between aquaporin 4 knockout. So long story short, severe effect was observed on the decrease of not angiotensin converting enzyme. This is an acetazolamide, the abbreviation of AC. Less decrease of drainage during anesthesia. So the acetazolamide and also anesthesia both are going to have an influence upon impaired the extracellular fluid of the brain, including everything, every waste products, those glymphatic, they call it glymphatic. Impaired glymphatic function was documented here because the patient was just 1 month after surgery. That fragile period, once given the acetazolamide, here extracellular fluid cannot be cleared very easily. That's C14 inulin. Inulin has a molecular weight of 4,000, and those inulin is not cleared well, meaning tissue fluids are collected, and those collected tissue fluids are going to increase the pressure, and that pressure is going to squeeze the vessel. What I'm saying is just capillary, and capillary, and then those small vessels are going to have a backflow. That's my current the interpretation after the incident, after 4 or 5 years. But there has been no consensus between the neurosurgeon, the professors, and ours. So that's my assumption. And here, child's nervous system. After this incident, let me tell you, we don't do dimoxepectin very young. Why? Because anyway, this patient is going to be operated. And we don't do children. Why? Because in our children, according to the report by my neurosurgeon colleagues in the various the journals, they say that with or without the diamond spect, they see the clinically almost complete improvement. So they don't need to choose the patients using SPECT. They don't need to confirm the improvement. Why? Because patients are improved. And they do just kind of continue to be in that state. So just like epilepsy, just like Dimox, just like epilepsy, in this case of young patients, we don't do this one. And we are doing more and more in elderly cases. In adult cases, interestingly, they could not find effect. Who would be improved after this kind of ECIC bypass or endarterectomy or stenting? So they send, based on their experience with the young children, I mean the younger age group, our neurosurgeon colleagues, they now know the efficacy or the contribution of diamond spect in adult patients. So after we see after 5 to 10 years, we can report this kind of in asymptomatic adult patients and symptomatic adult patients, and even with the intracranial hemorrhage complications, one might be the 10-year after outcome. Then we are writing the textbook. Please join us to rewrite and change the clinical routine using nuclear medicine. So that was the success stories. Then with just to kind of run in a fast way, you have listened to the progress of brain theranostics. In this case, theranostics therapy had been done and developed by neurosurgeons by the help of the neurologist and by the help of the radiologist and by the help of us, meaning the nuclear medicine physicians. And we've reached a kind of a plateau. I'm quite confident to share this information with you. And then I'm going to move on to, I have just covered only 10 the slides here. I have prepared, the 30. So we have remaining 20 slides. And then I'm going to resume after now the 1 o'clock. We have 1.5 hours now after 10 minutes. So 1:10, I'm going to start. And let me just show you what are remaining here. So many pictures and animations, and then some kind of the surrealistic, ultra realistic pictures. So you might just kind of seem to the peep over the future. This future is not going to be made by myself. Not by myself, but by the followers, meaning the next generation leaders of this department and my colleagues in this country. So the brain theranostics promises here, and then topics and issues, and patent their promises. And then you might just kind of expect better next hour, better quality. Okay, so let's come back after 10 minutes. Okay, welcome back, everyone. Unlike the first, first 1 hour, this second 1.5 hour is going to be a little bit rush. Why? Because the previous one was should have been the slow and steady, and to keep the meaning, the significance in everyone's mind. But now you're going to just kind of run and browse and then grasp some kind of notion. So let me just kind of make it a little bit easy. Sometimes I'm going to become a little bit extreme in expression, so please excuse it. Why? Because without that and without kind of posing myself as a devil's advocate to the against the entire scientific or medical community of this field, there is no kind of real understanding of this field. The pros and cons, or the bright and dark side of the current the one. Anyway, unlike oncology, we are not doing the many brain studies, meaning we are not going to be a help, and we don't have any the raison d'être, meaning the reason of the existence. So the promises of the brain theranostics had been there, Alzheimer's disease. I had been involved here. I had been involved in the clinical trial of the using fluorotaucipir for a one drop from maybe Pfizer. And after several years of endeavor, and they just told me, sent me and my colleague, a psychiatrist who are taking care of the Alzheimer's disease patients. They stopped their clinical trial, just like kind of bad news from out of nowhere. Okay, Alzheimer. In case of the Alzheimer's disease, let me just tell you. 3 years ago, in my room, I was sitting in my the chair on my chair, and I have invited my dear psychiatrist colleague, and he just tell us that once we adopted FDG PET, he was the one to use the FDG PET, and sooner he found that he told me that the FDG PET is just like MMSE score. Why should we do the FDG PET? And we concluded it's because there is no treatment of no treatment of Alzheimer's disease. Aricept is not working, and Aricept is not known to improve any FDG uptake. So if FDG uptake is improved, then the MMSE or the CDR is going to improve. So why should they do FDG PET? So that's the reason why we don't use FDG PET for Alzheimer's disease. And then amyloid PET had been invented by Chester Mattis and C11 PIB, and everyone was working very hard to adopt C11 PIB, PIB imaging for amyloid PET. And we just kind of assured ourselves that FDG PET had become obsolete because of the introduction of amyloid PET, and both of them are using radioactivity, so there is only one use. However, in case of the amyloid PET, there are too much positive findings in amyloid PET. So our Australian the pioneer and his colleagues and all the other people were trying very hard to use. Is there any way to just to improve? Once they adopted some kind of fancy statistical method from the statistics or the big data analysis or even deep learning by my junior colleague. And they have tried to use the amyloid PET. But according to my psychiatrist colleague's opinion, the amyloid PET, if anything better is going to be available soon, then they are not. He's not going to use amyloid PET again anymore. So the MMSE or CDR is enough, and still there is no the treatment. You know, 20 years ago, amyloid, the vaccination against the amyloid had been done, and after before finishing the clinical trial, they found that the even though amyloid plaque has been removed. So let me just take care of the issue. I just muted all, and then I just turned on mine. Where is it? So, we are waiting for tau PET. Then tau PET is going to be a kind of the breakthrough. The long story short, my just the interim conclusion is no. Anyway, tau PET, once it is available widely, then tau PET is going to kill amyloid PET. So in this kind of 예, 다시 올릴게요. 저거 하느라고. So wait a minute. So once tau PET is available, the amyloid PET will become obsolete again. Then what's the reason of this? And then actually pioneers, they would have their own situation and their own the objective and their own point of interest. But excluding pioneers in this field, every other, including us, we are the followers, and we feel a little bit the deserted or frustrated, the embarrassed to follow them or not. In the literature and in the textbook, they say that the amyloid PET is good and tau PET is good. Then what? Why we don't use amyloid and tau PET? And I'm not going to use the tau PET again. So we need to understand that. So the that failure of AD therapy, there's kind of CPR and strenuous cardiopulmonary resuscitation of that drug. The news had come from stock market and also Biogen and also FDA last year around September, in a small group of population, high dose of the aducanumab. The characteristics of aducanumab is the it's high, that monoclonal antibody is highly variable portion. The epitope is mimicking auto antibody, which had been reported from the humans. So there had been a story, but interestingly, until recently, until the April, just last month, there had been the March 30th in JAMA, Journal of American Medical Association, that journal, three committee members, the advisors to FDA, in the committee to evaluation, for the evaluation of aducanumab, its efficacy, they have written down that that drug cannot be resuscitated. Okay, so that's the small, scanty hope for the monoclonal antibody therapy for Alzheimer's disease. And I have, for the first time, heard neuroinflammation theory hypothesis for Alzheimer's disease pathogenesis 20 years ago. And recently, because of the failure of the clinical trial of using monoclonal antibodies, Eli Lilly, Pfizer, and many things. Here you see Alexander. This patient, these experts, they have given their ideas, evaluation of aducanumab, their futility, and around that time, since 2015. Just 6 years ago, there had been a huge discovery that, interestingly, brain has also lymphatic. So, the textbook knowledge, when I was the student in medical college, and since then, every textbook is saying that brain does not have lymphatic. But interestingly, 6 years ago in a Nature paper in humans and mouse, rodents too, there is a lymphatic clearance from brain. And then what about the amyloid tau hypothesis? In contrast with the neuroinflammation, amyloid tau hypothesis here, tau is working first, and then tau, A beta is working first, and then tau is following. And according to the current bold hypothesis, which was given by eminent a group of neuroscientists here, published in Neuron, peptide-dependent dysregulation of neural circuit dynamics in Alzheimer's disease. A beta dependent hyperactivities can be investigated using electrophysiology tool, classical one. And interestingly, tau is causing hypoactivity. And A beta is coming first, and then tau is following. And then once the patient's brain have both A beta and tau, then A beta and tau interaction is showing this kind of a hyperactivity or hypoactivity. And sometimes like this, and sometimes like C here. Tau dependent hyperactivity can match or synergistic, sometimes antagonistic, and it's phasic. And from time to time, from the beginning to the end, there are variety. So this has been just a kind of expressed in this kind of cartoon: neuronal normal activity and hyperactivity and hypoactivity, and rescue and no rescue, kind of very complicated. And in this another paper by the same group, they have said synergy between amyloid beta and tau. So tau is coming later. So what about saying that tau is a kind of a firefighter? Amyloid beta, A beta is a bad guy. Tau is coming to rescue, and then finally, they cannot match the tyranny and brutality of A beta. So whatever you just kind of keep this concept in mind, anyway, A beta and tau both are not going to help the neurons to work normally. Then, from those kind of features and results, the scientists have drawn two hypotheses. One is dual cascade hypothesis. The other is just a cascade hypothesis. A beta comes first and tau, and then neurodegeneration. A beta and tau both are, according to the GWAS, the genome-wide the association studies, many risk genes have been found in the pathway of the A beta metabolism and also tau. So, and we are not saying anything about the familiar one. We are interested in sporadic. And now we know that the neurons are not the sole player in brain. There are three more types: oligodendrocytes, taking care of the axons and myelins, and also microglia, resident macrophage, which is totally different from monocyte-derived macrophage circulating one, bone marrow-derived one, and they are different population. Just like copper cell is a resident macrophage in liver. Resident meaning not intern resident. It's resident, meaning from birth, they are already there. They don't need help from bone marrow or circulating monocytes. They rejuvenate there throughout the lifetime. In case of microglia, we know that their lifetime is half-life or lifetime is 4 or 5 years, meaning they are going to renew throughout the life. And as is expected, microglia is aging. So in the aged people, those microglia is not working very well to clear waste from the brain, interstitial extracellular spaces, even neuron debris, apoptotic bodies, or neuropils, or extracellular components, and also astrocytes. They are not working. Their primary role is the supporting and making a scaffold. However, they recently found to be the active player during sleep. According to astrocytes' role, based on the molecule aquaporin 4 and feed, they are active player to control and coordinate and organize the waste disposal from brain. And associated with a delta wave sleep, meaning the slow delta wave sleep, in the early part of the sound sleep, meaning the among 8 hours, the early 4 hours are desperately needed. If you sacrifice them, then waste cannot be removed. That waste includes adenosine. If adenosine is higher, you feel tired and you feel sleepy. And if you just kind of cut adenosine action using caffeine, then even though your brain's interstitial adenosine is high, you feel rejuvenated and they recovered from sleep, even though your brain's adenosine amount is high. You feel just kind of like in the morning, but it is actually kind of fake. Adenosine should be removed during slow wave sleep with a delta wave, the early part of the brain. So, the distal therapeutics, how to use the exercise, how to use the kind of physical and the mental exercise, and how to use the sleep-wake cycle to improve and to just kind of deter. So just to postpone the advanced introduction of the progress of the Alzheimer's disease. That's the concept of the distal therapeutics. Anyway, and we have many players here. And here, current PET imaging is like this: amyloid, fluorobetaben. We are doing that. Fluorotemetamor, that's being done by Asan Medical Center, and beta-peer, and PIB, and tau fluorotaucipir, and PBB. Okay, but interestingly, amyloid imaging is explicit, meaning those compounds are seeking beta sheet of the amyloid plaque. Amyloid is, according to the biophysical explanation, is once extracellular A beta is going to become the nucleus, meaning nucleation, and then elongation, and then fibrillation, and then aggregation. That kind of process, during that kind of process, after some period, we do see the beta sheet structure, the alpha helix and then beta sheet in protein structure, peptide structure. So those amyloid compound, amyloid-seeking compound is made of the plain type of structure. It's a completely planar structure, and benzenes, and they are and they cannot rotate and they cannot bend. So that's the situation. And amyloid imaging agents have that kind of characteristics, and they are quite similar to each other. And then what about the tau? Tau is an intracellular molecule. Actually, tau is related with the they call it MAPT. So it's kind of microtubules are phosphorylated and dephosphorylated very frequently, and tau is helping that. So that intracellular molecule, and pathognomonic, the status of tau is neurofibrillary tangle, and it's intracellular inclusion body. However, recently, tau had been found to be excreted via Golgi apparatus. No, tau is excreted in an unconventional way, meaning not using Golgi apparatus, but using heparan sulfate. So that's a kind of a tau can be directly secreted. However, however, tau has more ways to be communicated between the brain cells. Tau is not only the transferred from neuron to neuron, neuron to microglia, neuron to astrocyte, astrocyte to microglia. The problem, the difference is only the which one first and which one last. So that kind of tau. So then tau imaging. What are we doing with the tau? Tau is making, in addition to intracellular the inclusion body, which is called neurofibrillary tangle. Tau is also found in plaque, near plaque. So we call it neuritic plaque or dystrophic neurite. So, or tau is also found, according to the neuropathologist eye, as a neuropil. So tau has many forms. So what kind of forms are going to be imaged using tau agent? And then tau agent is also the seeking beta sheet. Tau is also taking the path from nucleation, elongation, fibrillation, and aggregation. So tau and tau and another the peptide, just like in case of the so let's forget it. So tau as a peptide propagating from cells to cells. Tau imaging agent for PET is also seeking beta sheet. Then amyloid beta sheet and tau beta sheet, it's all beta sheet. Then how those tau seekers for tau CPR and PBB can work. So we found several years ago by the help of the radiopharmaceutical scientist in this institution, I learned from him that, interestingly, first tau beta sheet is a little bit bent. So tau agent should have a bent, not just a planar. So that the degree of the bending is making tau agent different from amyloid. So once tau agent is invented or discovered, then we need to check that is there any kind of differential sensitivity for tau and amyloid. And then in that case, mostly the problem is the neuritic plaque tau, meaning extracellular tau. Sometimes it's a truncated, sometimes it's many kind of post-translational modification. We call it PTM, post-translational modification, including phosphorylation and many kind of other the modifications. So among 440 amino acid, the serine or threonine at this point is phosphorylated. And this antibody is taking care of this one. And however, there are much more. Here, phosphorylation sites are so diverse. So once people thought that the tau post-translational modification is the key issue of the abnormality of tau, which is found to be not the case now. And, so the immunohistochemistry cannot reveal the abnormality of tau. And then, this too, in case of amyloid plaque is a pathognomonic finding of Alzheimer's disease. It's correct. That's pathologic signature. However, it is a different story whether amyloid plaque is making things worse, or is there another culprit? The people, meaning the scientists in the field of Alzheimer's disease and neurodegeneration, they now think that A beta oligomer is the culprit. And then what about the amyloid plaque? Amyloid plaque might be a good source by the help of the microglia to release A beta, but the A beta produced from the amyloid precursor protein on the membrane of the neurons, APP, is a more correct source of A beta. So considering that kind of knowledge, what are we doing now? We are working on the PET ligand and PET imaging for beta sheet of amyloid plaque or beta sheet of the extracellular elongated and fibrillated and aggregated tau. Are we taking care of the, are we concerned about intracellular inclusion body? No. I have found no one who is explaining this issue clearly. You have just heard my explanation. You might be confused. So it is not your own confusion. There had been a long time confusion within the Society of the Neuroscience, within the Society of Neurodegeneration Research, and also the therapeutics investigation. That's the kind of a lack of knowledge. With that kind of, despite that kind of lack of knowledge, the pharmaceutical companies poured a huge amount of money and developed monoclonal antibody to anything, meaning the amyloid and tau. Now they found that amyloid is hopeless, and then they just turned into tau. And we are still waiting for the correct therapeutics. Once therapy is available, then we are happy to use our PET, amyloid PET or tau PET, to find the efficacy of the therapeutic effect of the as a surrogate marker, in addition to the behavior. So tau and amyloid and their imaging, in retrospect, they have been kind of in the wrong direction, in the wrong realm from the beginning. So the kind of failure of a therapeutic trial were doomed. And still, until very recently, they just confess that, the scientists confessed that in 2019, there are other ways of exchanging tau, either truncated, either mutated, either phosphorylated. Those taus, that kind of modified taus can be a nucleus for the further nucleation and elongation and fibrillation and aggregation. However, so once it is transferred from a diseased neuron to another cell, then it is going to be the one mechanism is exosomes, the other mechanism is the tunneling nanotube like this. All these cannot be studied in vitro very well. But in vivo, things are, so you see the reddish one is going here, and then, so as I just tell you. Heparan sulfate, unconventional, ectosome or exosome. Please don't be overwhelmed. There are so many the names of the same, similar. And tunneling nanotube is beyond our understanding now. And they tried very hard to find out if there is any kind of sequence specificity, or any kind of structural characteristics here for developing therapy or for understanding pathogenesis. And crude conclusion is no, there is no clear, simple understanding. So that's the reason why this woman doctor from in the States, in the United States, from but originally from Taiwan, that Virginia Lee, 10 years ago, she proposed peptide propagation hypothesis. Once tau is out, just like prion, it's propagated. And one neuron and one microglia and one astrocyte is down, then the nearby cells are being affected. Even though there are so many versatile mechanisms is working to eradicate neuronal dead body, apoptotic body, or the neuronal debris. So once they are dead, if not eradicated, those debris or peptide themselves, they propagate to other area and other cells. So AD-like neurofibrillary tangle or neuritic plaque tau or neuropil thread. Neuritic plaque tau is propagating like this. Fibrillary tangle is the intracellular one, so they seemed to take transsynaptic, the transfer, and neuropil thread is kind of very important from the eyes of the from the standpoint of the neuropathologist. But anyway, the so many diverse. And until recently, molecular psychiatry here, sequential formation and different layers of dystrophic neurites in Alzheimer's brain in human. Post-mortem brain had been an amyloid core, vesicles, mitochondria, and endoplasmic reticulum tubular inclusion, and also autophage vesicles. Autophage is kind of a recycling machine. In addition to smolation and ubiquitination, they have another mechanism. The discoverer of that phenomenon, a Japanese scientist, won recently Nobel Prize. Meaning, we don't know exactly how the remnants or the excreta from the dead and disabling diseased neurons and cells, brain cells, are being taken care of. You know that everyone is saying the neuritic plaque here, AD, senile plaque or neuritic plaque, or the amyloid plaque. We call them like this. Neurofibrillary tangle is Alzheimer's disease. Both of them are the characteristics. But you know, the and the significance of the intracellular inclusion and extracellular tau are recently being discovered. And if I show you a little bit, that's the conclusion. Neurofibril tangles are not required for neuronal silencing. It's reported in Nature Neuroscience. As I told you, using calcium imaging, hyper excitability, the fractions of cells which is excited had been counted. That's counting. So this field. Let me just take care of the audio issues. Sometimes, interestingly, some breakthrough is happening. Okay, so I had a hard time to let you just kind of have a glimpse upon the complicated issue of tau, amyloid and tau. Amyloid has two problems. One is the amyloid plaque. Is it really detrimental to the patient? Found too much early. Some of the normal persons are showing the amyloid positivity. It's actually amyloid positivity is not a correct term. Amyloid plaques beta sheet positivity. But interestingly, some of them are normal or MCI, mild cognitive impairment, and non-converter. So do we need to just find the MCI mild cognitive impairment patients with with a possibility of least possibility of conversion? Should we find them? No. So this field is really beyond our understanding so far still. And then that's a biophysical term. Amyloid self-assembly and molecular strategy for interference. There are so many excellent pharmaceutics experts to find out the remedy here. Non-fibrillar species, nucleation, elongation, and oligomer themselves are toxic, fibril fragmentation, fibrillation, and finally making an assembly. Here, Angewandte Chemie 2020, applied chemistry. Then what about now for tau? Tau again, there are so many efforts that same nucleation, elongation, and then beta sheet formation, and finally contribution to the neuritic plaque. Now for tau realistic, meaning in vivo. Nucleation-dependent polymerization has been used to prove the purpurine type of drug is working. And again, another person in terminal capping amyloidogenic tau fragment. So still they are working on, they are peptide chemists. So that's their expertise, but I don't think it is going to be successful. Why? Because there are so many players and there are two culprits. They are in synergy and sometimes antagonism. And so heterogeneous results. That synergy is for what? Pathologic aggravation or they are on themselves. They are trying very hard to compensate the other guys' ill doing. So still, tangle containing and tangle free cells. Interestingly, for the first time, 2020, they are working the same. So the interim conclusion was neurofibril tangle or aggregates. The final waste product as a neurotic plaque does not seem to be the current current agent of toxicity to the brain. Then why should we image those findings to prove the patient has a Alzheimer's disease? Really? Alzheimer's disease is the disease without proper diagnosis. They are going to progress more rapidly? No. Alzheimer's disease does not require a brain biopsy for diagnosis. Then why should we develop amyloid and tau imaging? Only when they are related with treatment. If treatment is available, then we are happy to advertise our PET studies are going to help. Since 20 years ago, my conscience told me that I would rather not advertise the efficacy of amyloid or tau imaging in the media, meaning when I was invited to say in public on the newspaper or television, I cannot, I just kind of conceded. I just try to withdraw, not respond to that kind of suggestion. We need therapy. Then what kind of exosome issues? You know, exosomes once in the interstitial fluid, they are working like this. However, sometimes exosomes are, that's a fluorescence labeled exosomes. They do circulate. So inter-organ communication was proven in this zebrafish, but we are not fish, okay? So, the problem is there are further parts of the production of eradication on site and then the release, clearance from that site to the systemic, the milieu, meaning the lymphatic drainage. So those issues, we've entered into that field 2 years ago, meaning my colleague, Professor Y. Choi. She had done this kind of study and she found that the lymphatic drainage can be just kind of, can be obliterated by lymphatic, the ligation, lymphatics ligation just before the deep cervical lymph node. And then after ligation, we measured in a human familial Alzheimer's disease gene transgenic mouse model here. And then we used the focused ultrasound to enhance the clearance, not just to open the blood-brain barrier, but enhance the clearance by influencing astrocytes. And we measured CSF A beta human A beta. Why? Because this animal has a human A beta, the abnormal mutated A beta. So that A beta is working as a nucleus for elongation and nucleation, elongation and fibrillation and aggregation. So we found that the CSF A beta is really high here. So they explained it and we published it last year. And since around that time, one of my colleagues from this country, they just did a thorough study about the lymphatics. They found that here, as I just draw the cartoon on the right side, mouse, the superior sagittal sinus blood flow is from front to from anterior to posterior. And the 6 years ago in a Nature paper, they found the mouse has a dural lymphatics, and also the nasal cavity flow of the interstitial space here, nasal cavity or the lymphatics are leading to the anterior to posterior. And they have the same direction from anterior to posterior, blood and lymph. But interestingly, according to the MRI study, these are human lymphatics structure here. And superior sagittal sinus blood flow is from anterior to posterior, just like a mouse. But interestingly, lymphatics here, blood in the superior sagittal sinus is white, then it's black. That blood is black, and then here, lymphatics are showing white, meaning the counter current flow. So the aging process of lymphatics of mouse is different from human. It's because of the direction. So that kind of issues have been recently elucidated. So based on that, we could just say ISF to interstitial fluid to CSF to perilymphatic space, meningeal lymphatics, and then sub-lymph node. That, we have published the fourth edition of nuclear medicine textbook in this country since 1992. The first 3 editions said humans do not have any lymphatics from the brain, so CSF should be diverted. I mean, should be cleared from the CSF space to superior sagittal sinus vein via arachnoid villi, which is incorrect. Now the correct knowledge is only 10% can use that pathway. CSF via arachnoid barrier cells directly to meningeal, meaning dural extracellular space, and then dural extracellular space has a really spider of lymphatics. So the lymphatics has a really so many holes. So once CSF is going through the arachnoid barrier cells and dura, and then dura has a lymphatics, and within the lymphatics, and lymphatics are coming down. Humans do not use nasal cavity. It's against the report in the 2 papers of JNM, which is wrong. And here, mouse and rats basal meningeal lymphatics as well as nasal mucosa. but humans, meningeal lymphatics all over the cranium and drainways without the help. So here, classical 24-hour brain CSF RI cystosonography imaging is showing that the all over the CSF space is filled with indium calcium TTPA, and those the TTPA is being removed by capillary? No. By superior sagittal sinus? No. They are removed slowly to meningeal lymphatics and meningeal lymphatics to deep cervical lymph node to thoracic duct and subclavian vein. Okay, that was our classical imaging, and then recently the MRI people, they have used the intrathecal injection of gadobutrol, and they imaged the same one, 26 hours and 50 hours. So wouldn't it be interesting? We do first, and then MRI people is doing the following, and then they just lose interest. Still, we are doing the RI cystosonography to help the communicating NPH, communicating hydrocephalus, normal pressure, to find out if there is any reflux here, ventricular reflux. If there are ventricular reflux, then we see that the ependymal cells are activated to clear CSF. Why? Because of there is a basal cistern obstruction or the problem of the lymphatics of now. My just interpretation, problem of the meningeal lymphatics. Then here, VP shunt, ventricular peritoneal shunt should be done. And then that patient is improved. If there is no ventricular reflux, then there is no way. Reflux should be there, okay? So recently, we've done albumin labeled with copper 64. That's Dr. Asmar's work last October. Then the intrathecal injection of albumin. We are not using albumin in humans in intrathecal use, but in animals, we've according to the approval by the IRB, animal experimental IRB. Once injected, those are reaching the here, and then both nasal cavity and also the meningeal lymphatics, they reach superficial the lymph node, and then deep cervical lymph node. Superficial cervical lymph node, which is also called mandibular lymph node, is from nasal cavity. Okay, and the people, the investigators so far, they have used the C14 labeled, iodine 125 labeled, A beta or inulin, or tau or albumin, whatever, or dextran or fluorescent dye themselves, even quantum dot, they have injected here. And then here is a striatum. Here the optic bulb, olfactory areas, and striatum, and red hypothalamus. Green is a hippocampal region. This image is from Allen Brain Atlas. Now, I have one or two issues. We have run down the most difficult part. And then I'm going to show you several images. But before going into the a little bit futuristic ones, I'm going to ask you here, barrier anatomy. Why? Because once you are going to inject blood, the monoclonal antibody for a remedy, you and me, we used to think about the crossing blood-brain barrier. However, in normal cases, how the amyloid is sensitized to the immune system. So we need to think about the what? Very difficult subject, neuro-immune interface here. Neuro-immune interface. How the immune system is communicating with the brain by just the crossing BBB. Then why so many microglia are there? Microglia, so many. And microglia is totally different from monocyte-derived macrophage. Monocyte-derived macrophages from bone marrow. And that microglia is residing here since birth. Before birth, it's coming from where? Not bone marrow, yolk. Sac. That's bone marrow. Bone marrow is a central for immune system. Brain is central, brain and spinal cord is central to the neural system, but immune system, the centrality resides in bone marrow. So that neuroimmune interface. So let me just ask you, who? Dr. Charisma, you can respond. You're one of the person who asked me about the amyloid and tau PET. Yeah. So let me ask you, choroid plexus is within the brain or outside of the brain?
Sorry, plexus in?
Yeah, choroid plexus. You know, the choroid plexus.
Yes.
Choroid plexus is within brain barrier or outside of the brain barrier? And then Andreas, you can just respond to the next question. Ependymal cells of the ventricle, does it have a tight junction or not? So meaning CSF is within brain barrier or outside of the brain barrier? These two questions, no one they succeeded in answering these questions recently. It happened in January with my fellows, and before around that time, before that time, I did not realize that too. So nobody taught me, and no textbook is easily saying. Dr. Charisma first. Choroid plexus, is it
in the brain? It is barrier for the brain.
So the choroid plexus is within the brain. Yeah. Then where is the barrier? So that's following question. And Andreas Lim. So can you respond? Yeah. Okay.
Yes, bro.
Yeah. CSF is within the brain or outside of the brain? Meaning the ependymal cells are within the tight junction or not? Just a guess. I did not know until December.
Outside the brain.
Okay, so Dr. Charisma, the choroid plexus is within the brain, correct?
Correct.
And then the Andreas Lim, you told me that the CSF is outside of the brain, correct?
Yes, bro.
It's contradictory. Both of your answers are contradictory, okay? Right, so this had been not so important so far. So then the spin-off question to Dr. Charisma. If you think that choroid plexus is outside of the inside of the brain, yeah, inside of the brain, then where is the kind of a tight junction between choroid plexus and brain parenchyma? So it is not easy to answer that. Why? Because we used to not to think about the brain is how brain is protected with a tight junction from the system, meaning outside of the brain. So thank you, Andres, and thank you, both Dr. Lim and Dr. Charisma.
Thank you so much. Thank you so much, Prof. Lee. It is very interesting.
Yeah. So this is the kind of answer to that question. Here, our Japanese colleague, he's a neuroanatomist. He has written down in Neuroscientist journal just 2 years ago, 2019. And he just had written down in his kind of a review paper, in the introduction, he found that the big shot investigators, best investigators in the world, they don't understand this point. So he was the one who before me who was trying sincerely to just explain this. Let me just say, everyone is only interested in this one. This one meaning BBB, blood-brain barrier. But there are two more important barriers. One is here, dura, arachnoid, and pia here, and yellow is a CSF. Dura has this layer, arachnoid barrier cell layer. This has a tight junction. So no macromolecules can easily pass through easily. And again, here, choroid plexus is here. Around the choroid plexus they are filled with the CSF, and the CSF is within the ventricle, correct? So we say that it's a brain CSF barrier. Okay, brain CSF barrier is here. Epithelium of choroid plexus has a tight junction. Here, capillary are coming from the system, choroidal artery here, from the internal carotid artery system. But interestingly, here, according to Nakata, is a good explanation that endothelium does not have tight junction, has a gap junction. Meaning these areas are outside of the brain. Outside of the brain. Choroid plexus here, stroma, is the best candidate, one of the best candidate of neuroimmune interface. Tight junction is here. And ependema, you're here, ependema in this cartoon, in his paper, Nakada, clearly saying that gap junction. So contrary to Dr. Lim's answer, CSF space is inside of the brain. Why? Beneath dura, arachnoid barrier cells are covering CSF and brain. PIA does not have any tight junction, right? Here we know that brain endothelial, capillary endothelium has a tight junction. So parenchyma and CSF is inside. Choroidal plexus is outside. Now, the 50 people from 30 different sites, today, you are the first person to recognize this. It's, this fact has become a problem since 2005, 15, not 5. Here I have written. In Nature paper, brain has a meningeal lymphatic clearing ways. That ways include adenosine, lactate, small molecule, abnormal protein, and possible. Oh, excuse me. Let me just handle mute situation again. Now, let me. Okay. Okay, sometimes one of you just unmute unintentionally. So I'm taking a break, and everyone they simultaneously take a break. So let me just I should not forget to share again. Why? Because to handle those mute situation, I need to. So brain has a meningeal lymphatics, and brain has waste clearing system, lymphatic, that kind of issue. So that's brain anatomy. So if you are going to use the monoclonal antibody, so you need to pay attention to the choroidal plexus stroma, and also the meningeal here, dural lymphatics. Then let me just ask you a short question and then answer by myself. Then until recently, there had been a real puzzle how CSF can just cross this barrier. Why? Because the arachnoid barrier cell layer is not well known, well characterized. This is very difficult area, just a thin layer. And you know, here, subdural hematoma here, subarachnoid hemorrhage here. But like this. Interestingly, our anatomist colleagues in the world, from Finland, from Rochester University, United States, and from Japan, they just they independently tried very hard, and they found that there is a fissure here, fissure. Lymphatic endothelium tunnels are the crossing the fissure. And the functional, physiologic, and pathophysiologic roles are still to be delineated, meaning they discovered. And then now, you know, you have encountered, you have listened to the recent progress, which was digested by myself last year by the help of the COVID-19 crisis. Without COVID-19 crisis, I would not have time to sort out those weary the informations. So Next semester, if you just can just bear the kind of alien language from the computing, statistics and mathematics or geometry, then I would just tell you about the PET fMRI and topography with AI and deep learning, and topology and hyperbolic geometry. Topology is kind of geometry, topology, and algebra are the three, and analysis and then differential equation, and also the those are the computing issues are, and then okay. Once again, tau PET will become obsolete while both amyloid tau PET struggles to become surrogate imaging biomarker of novel treatments. Now you understand why I have written down this kind of sentence. This sentence is not from the literature, it's from my mind. I don't have any conflict of interest of this kind of a criticism or the sarcasm. Why? Because it's for you. You should, you might not the waste time. Please don't, please just survey the literature on your own, by yourself, very eagerly. And please find the best the colleague among the scientists. Not the best scientists are not going to help you. We need kind of a small group. And then while just talking every day with them, you might find from the instinct of the physiology, from the instinct of the physician, and from the on the instinct of the clinician, you can just raise the correct question. And that correct question, some of the questions are not answered or are just kind of ignored. That ignorance has led us to the global failure of a new treatment. And then, so my just kind of suggestion is with this one slide. All the other slides are just kind of a corollary. New targets based on spatial transcriptomics. Do we know brain well? So brain should be viewed as a total. Molecular biology just kind of did a very good job, but it needed 50 years to accumulate that kind of knowledge. That knowledge is going to be open to everyone if you have a computer and if you have a correct mind. So it has become the available to have I mean, the spatial transcriptomics has become available in my department since November in my hand. Actually, my junior colleague, Professor Hong-Yun Choi, he just adopted this new technology, which is not so expensive, actually $20,000 per session. It is not so expensive. It was 100 times more, but recently and 2 years ago. So new targets here. If I if I remember the progress, when I first started the nuclear medicine, muscarinic receptor imaging was the kind of a hot topic, which was led by NIH chemist, radiochemist Eckelmann. It failed due to the demise of the cholinergic hypothesis. You might not know if there has been this kind of hypothesis. FPCIT is struggling due to neurologist consensus. Neurologists say FPCIT is not needed for routine evaluation of Parkinson's disease. And this point is going to be explained by Professor Kim next semester. I asked him, and he's the best expert here. And he's with the best Parkinson disease expert in Asan Medical Center. And amyloid PET explained, tau PET, yeah. The time will tell. And microglia, TSPO. I have heard TSPO imaging, you know, since 2 decades ago. Here, interestingly, in this science paper last year, Berlin, Freiburg, 2 eminent scientists wrote down in Science, we need PET ligand for P2RY12 and TREM2. So they said, thus the development of new radioligands for PET-based monitoring of microglial activity in the human brain isotope-labeled ligands with P2RY12 and then TREM2 receptors will be of importance. It's kind of begging to the world. And they explained microglia is a heterogeneous, just like a neuron. Every microglia is not the same. And astrocyte was imaged C11 deuterium L-deprenyl as a kind of a ligand for MAO, monoamine oxidase B, as a surrogate, and oligo and non-neuronal non-glial cells. And recently, immune cells are residing there in addition to microglia. That's called border-associated macrophage. Interestingly, perivascular macrophage is also residing there from birth. So it's just like its characteristics is macrophage, but it is not monocyte derived. It's unrelated with the bone marrow. That kind of issue should be understood if you are going to use the monoclonal antibody for a brain, not just opening BBB. If you open brain, the BBB, sufficiently, appropriately, then monoclonal antibody is coming into the brain. No, everything is coming into the brain. So, we need more knowledge here. So this is a kind of an introduction. If you also request or ask or allow me to do, then spatial transcriptomics for brain is going to be explained. Spatial transcriptomics is a kind of a futuristic one. The current one is single nuclear RNA sequencing. If you just collect a nucleus, then mostly oligodendrocyte and neurons are there. So we need to just deprive these two, and then you find the astrocyte, microglia, oligo, and those things. Single cell RNA sequencing is giving you the oligodendrocyte are the many, and then astrocyte is the next, and neurons are many, however, different types. This is the data we acquired several months ago, and I have repeated the evaluation myself. You know, I learned how to analyze this one. Neuronal marker here, except for white matter, there are even distribution of pan-neuronal marker. And then what about the myelin basic protein is oligodendrocyte marker, my MVP is the reverse image of this one. And HEX-B is a common core marker of microglia. And then here, P2RY, HEX-B is increased here. 7 months Alzheimer's disease mouse model, they are showing increased gene the expression. But you know, though we are seeing the gene expression, not the number of microglia. That gene expression can be can represent the increase of the microglia increased number of microglia, or increased expression per microglia and whatever. So interestingly, P2RY12, which was just kind of summoned, invited by the best neuroscientists, unlike HEXB, those markers are rare. So if you find a ligand, then images is like that. It does not follow the regional distribution. It does not have the photon flux of just like FDG, then you are doomed. You are going to be fooled by the signal, low signal to noise ratio. However, in case of astrocytes, we have two specific astrocyte glutamate transporter here, and also astrocyte is known to have exclusively expressed glutamine synthase. I have made this image. You can find easily hippocampus, and everyone knows that SLC1A2 is EAA2 is glutamate transporter GLT1, has a 75% of propensity and 25% EAAT, excitatory amino acid transport 1. So this kind of information is in our hand. So if you are interested in TREM2, interestingly, TREM2 has many. Along the aged the Alzheimer's disease model, and GFAP2. That's if he paid the $15,000 to the 10x company and Macrogen, which is a sequencing company, then you are going to receive this kind of image here. This is the last slide. We have listened to the TSPO imaging as a neuroinflammation marker. These are others. However, TSPO is so scanty. How can he designate this kind of material as a target? So this is going to be failure. If you're going to use this kind of SNAP25 or myelin basic protein, then you are going to receive the FDG-like image. But if you just follow TSPO imaging, as you just see in the textbook, TSPO imaging is really miserable. We cannot delineate any anatomy. The signal to noise, the ratio is doubtful. We cannot be confident that this is an increased and this is a scattered, this is decreased. So this kind of issues should be checked, should have been checked in advance. Okay, here we have several thousand spots, and each spot is giving the yielding 20,000 mRNA. Those mRNA, that technology was first reported here. No, not this one. Reported here, Science 2016. Yeah, here it was it was really interesting 5 years ago. Spatial transcriptomics, tissue sections. And what kind of technology they have used? SPOT has this kind of tissue section, and mRNA is captured by this here, tissue permeabilization. And then they have established, and they have compared in situ hybridization. And because it's spatial, I think this should be used by nuclear medicine physicians when we are going to choose radiopharmaceutical for targets. But keep this in mind. These targets should be peptide instead of the mRNA. We are using. We are kind of. I don't think the spatial proteomics would be possible. Spatial transcriptomics is possible because they are taking care of the sequence. The sequence can be amplified. But peptide, in case of peptide, there is a limitation of monoclonal antibodies against epitope. So if the spatial proteomics is not possible, then we need to be satisfied. We need to harness, use, and exploit and take advantage of this technology not to lose our lifetime. I lost my lifetime to follow the rainbow. That rainbow was the common consensus of neuroscience field. I had been Society for Neuroscience meeting several times in the United States to find the current progress, and I was really happy with the Chester Mattis success in C11 PIB 20 years ago. And 10 years ago, we've finally adopted fluorobetaben. I have been involved in using fluorotaucipir for clinical trials, which was interrupted, which was given up abruptly by the Pfizer company Pfizer. Was it Pfizer or Eli Lilly? I forgot. Eli Lilly. Eli Lilly. So next generations and the physicians, nuclear medicine physicians, when you start your career here now, spatial transcriptomics is available. And you now heard the story of loss of lifetime. I really, I'm disappointed with the entire neuroscience and Alzheimer's disease research field. They have pursued the wrong direction, and we were behind. We, meaning the nuclear medicine physicians, majored in the brain. We were behind. So now we have more than 4 countries and more than 30 institutions and more than 50 people, next generation leaders. I don't think every one of you would make a difference, but at least, hopefully, one of you is going to make a difference. In our future of nuclear medicine. And so as was promised, I think I don't have, well, because I just invited you to the future. So I'm not going to just bother you to ask questions. You please send your advice and request to Professor Soo. And Professor Soo is going to find the correct person to give you the lecture, easy and merry, easy and quite understandable, yeah, comprehensible from your standpoint, not the lecturer's standpoint. And then next semester is going to be announced soon, maybe within several weeks. And then today I'm going to announce to you that this summer we are going to hold another radiochemistry session. But in this summer, as we have collected experiences regarding the summer and winter workshop, previously we did it's country-wise, maybe one country. But for this occasion, we are going to hold 2. And then you can just kind of log on anyone. So it is going to be a radiochemistry. And I have a plan to invite senior, and maybe those seniors are going to be a little bit like me. So if you need a on hand, not cutting edge, not futuristic, on hand experience is in radiochemistry 2, radiopharmaceutical science 2, then please let us know. We can just hold another third one, and then that third one is going to dedicate it to the practical issues like technology, right? Cyclotron or whatever issues. So, or the easy way to the label the technetium compound and whatever. And Professor Soo Lee has been promoted to clinical professor recently. So he has a much more interest in doing the clinical expansion of radiopharmaceutical science and radiochemistry. So that time issue would be any week, Wednesday noon is going to be the time. But if we choose and then collect the possibility of participation, attendance, then we will choose 2 or even 3. Okay, that's our current plan. Okay, thank you for your persistence and sincerity to attend. I wish every the occasion would be a real help. Not currently, but for your future. Current one is you are already experts. So, okay. See you next semester.
Thank you very much, Professor Lee.
Thank you.
Yeah.
Looking forward to having you all listen again.
Yeah,
Thank
nice
you
to
very much.
hear the Hendra. Okay.
Thank you very much.
Thank you. Bye. We will close. Yeah. So not we will. Manager will close.